Okay, first chapter is cell biology. We're going to start with the cell membrane. We know it's a lipid bilayer. It has proteins, it has enzymes, it has receptors on it. The cholesterol increases the membrane fluidity. That's the most important thing you should know about cholesterol. And cells that are usually, because of the sodium-potassium pump, sodium-potassium is 3 out, 2 in, which means 3 plus out, 2 plus in. 2 plus in is less than 3, which means it's going to be more negative on the inside. And the sodium gradient that creates by pushing sodium or pumping sodium out of the cell creates a gradient for co-transport of sodium and glucose or protein or other molecules into the cell. When you talk about electrolytes in general, you want to know which of the electrolytes are usually in the extracellular matrix, and that's NaCl, right? You have NaCl, and then you have calcium bicarb. So just memorize it like that. And then in the intracellular, you have potassium, you have magnesium, and you have phosphate. Obviously, you have sulfate and you have HPO4, which isn't that phosphate? I don't know. Yeah, no, phosphate's on the inside, right? So yeah, sulfate is also extracellular, but it doesn't really matter. Just remember NaCl, calcium bicarb, and then potassium, magnesium, and phosphate intracellularly. Okay, so if you remember anything from histology, you remember desmosomes and hemidesmosomes. And desmosomes are between cells. Hemidesmosomes, because it's half of it, is between the cell and the basement membrane or the extracellular matrix. Tight junctions, you just have to know they're occludins, they're impermeable barriers. Gap junctions are for communications. They're called connexin subunits. Not really important for you to know. There are 2 types of receptors in the cellular membrane. And there's also the G proteins and the ligand-triggered protein kinase. The main difference between them you just have to know is that G protein is the one that usually creates CAMP, CGMP, and all those stuff. Inositol triphosphate, I think, as well. And the ligand-triggered protein kinases are a single transmembrane protein, but they have the receptor and the response enzyme, which means, and also they can be like a ligand triggered ion channel or a ligand triggered protein kinase. A protein kinase usually causes a release of an enzyme, and the ion channel usually allows the entrance of a certain enzyme. Like the ion channel would be like in the nerve muscle junction where calcium binds, and it allows for the other calciums to enter the cell. Sorry, I mean the acetylcholine binds, which allows the calcium to enter the cell. Anyways, so let's just move on from that. You have the ABO and HLA type antigens. The only thing you should know about these for now in terms of cell biology is that ABO antigens are glycolipids, and the HLA antigens are glycoproteins. Right, and then you have the osmotic equilibrium, which really doesn't matter. You have the cell cycle. So G1, S are together, and G1 we know is S is protein synthesis, obviously, because S synthesis, and G1 is the chromosomal duplication. So that's the first step. And then you have G2M. M is mitosis, so G2 has to do with the nucleus divides. So G1 the chromosomes duplicate, G2 the nucleus divides. In like general terms, the most variable one is G1, and this is the one that determines cell cycle length. What does that mean? So if you have a long G1, you're gonna have a long cell cycle. It means the turnover is gonna be low. It's gonna take a lot of time for the cell to duplicate. On the other hand, a short one makes sure that the duplication becomes quicker. And what affects that are growth hormones, growth factors in general, including growth hormone, obviously, which decrease the length of G1. And obviously, as you all know, G1 can turn into G0. We'll try to read that, quiescent, I think, which means it's just regular, it's not dividing, which is the case for a lot of the cells in the body. So mitosis is, just remember, prophase, metaphase, anaphase, telophase, right? Prophase is like the beginning. So you have the centromeres attaching, the centrioles and the spindle formation. Basically, spindle formation is that's where the thing that's going to be on polar sides of the cell, that's going to pull the chromosome. It's going to connect to the chromosomes using spindle fibers, and then it's going to pull them away. But let's just talk about prophase. Yeah, you have the nucleus disappearing. That's the most important part of the prophase. That's how you differentiate it. And then you have metaphase, where everything just aligns. When you're meta, you're aligned. You're in like Zen or whatever the hell that is. Anaphase, everyone's selfish. Everyone is like ana, just for themselves. So you have that. So their chromosomes are pulled apart. You have telophase, is where it starts to divide again, like it's starting to separate. But the most important part of telophase is that the nucleus forms around the new chromosomes. Since they're pulled apart now, كل واحد على جهة, like each group is on each one side. So you have a circle, so like in the nucleus starting to form around the first one and the second one, and after that the cell can divide into two cells. And you have the nucleus transcription and translation, right? So the nucleus is also a double membrane, which with the outer membrane attached to the rough endoplasmic reticulum, and we'll talk about why that's very important, actually right now. So the nucleolus is inside the nucleus, there's no membrane, and ribosomes are made here. So ribosomes are made in the nucleolus, and then they go and attach close to the rough endoplasmic reticulum. Why is that important? Because after transcription is where like any sort of signal comes, tells the nucleus, hey man, create this hormone, like I want this hormone, or I want this protein. But how does the nucleus do that? It takes the nucleus, it takes the DNA itself, or RNA, and, well, in our case, DNA, and then it cuts it from a certain part of it. It takes a certain code that it wants from it, and that's RNA, that's DNA transcription, creating mRNA, messenger RNA. So when this code is taken from the DNA, and duplicated into mRNA, the mRNA then travels into the it leaves the nucleus, and it goes to ribosomes to create like the protein. But we'll talk about it now. So basically, we talked about how transcription is created. But what are those factors that tell the nucleus in the first place, hey man, create this protein for me? There are two main types. Let's talk about them. So there's the steroid hormone and then the thyroid hormone. The difference between them is that the steroid hormone binds in the cytoplasm, right? And then it binds to the receptor in the cytoplasm, and then the receptor takes it to the nucleus and escorts it to the nucleus, right? But on the other hand, when you have the thyroid hormone goes straight to the nucleus. It does not need any sort of escorting. No receptors in the cytoplasm. Initiation factors are exactly what they are. They are initiation factors. DNA polymerase chain reaction, you don't have to really get to know that too much. To be honest, this is more like step or basic than you need to know. You have purines and you have pyrimidines. All you need to know is purines are A and G, and pyrimidines are C and T, and U. Well, T in DNA, U in RNA, but let's just stick to T, right? So you have A connected to T and G connected to C. A is adenosine, T is thymidine, G is guanine, and C is cytosine. So between A and T is double, between G and C is triple bonds. And you just need to know that for that, to be honest. I can't imagine how they can ask too many questions about it, and at least for the ABSITE review, right? Anyways. And then you go to translation. Translation, as we said, is when the mRNA template or code that's taken from the nucleus travels to ribosomes, right? And then a protein is created. So you have ribosomes. They're a small subunit that reads mRNA and then binds appropriate tRNAs that have amino acids and eventually make proteins. So, okay, basically mRNA is the code, the template. tRNA is transport RNA. And they basically, if you want to think about them, they transport the amino acids to the ribosome, so then it can create the protein, right? So it's like the code is there, the mRNA is there. The ribosome is like, hey, I need tRNA, I need this, I need alanine right now. Goes, brings the alanine, binds alanine to the construction site, and then asks for the next one, and etc., until it's all done. And usually, actually not usually, I don't think this really matters, but if you want to read about it, if you want to know about it. The opening, so you have the ribosome moving, and then a tRNA comes and opening the site, the A site for the next amino tRNA complex, basically amino acid and tRNA complex comes and binds at the A site. The P site is where the protein is already being made. A site is when the new tRNA comes and it's like, hey man, here's this amino acid. Anyways, this is a bit too confusing at times. It doesn't really matter. I just need to tell you this for the sake of telling you it. You probably know it already. And then you have the cellular metabolisms, right? So this can go into a whole detail. If you remember the Krebs cycle or not really important. The only thing I need you to know is glycolysis is the cytoplasm. It happens in the cytoplasm. It's one glucose molecule creates 2 ATP and 2 pyruvate molecules. And that's not really it's not really efficient, right? That's why we need the mitochondria. And this is where the mitochondria comes in. The mitochondria has the Krebs cycle in the inner matrix. The Krebs cycle doesn't create ATP directly. What the Krebs cycle creates is NADH and FADH2. NADH and FADH2 then enter the electron transport chain and they create an H plus gradient. So all you need to know is glycolysis creates 2 ATP and 2 pyruvate. 2 pyruvate go into the mitochondria, go through the Krebs cycle, create NADH and FADH2. NADH and FADH2 create a gradient, an H plus gradient, and that gradient is used by the mitochondria to create ATP from ATP synthase. ATP synthase is usually on the outer matrix, outer border, if I'm not mistaken, but it really doesn't matter if you just know it or not. Totally, one glucose molecule creates 36 ATP molecules, or ATPs, yeah. That's the most important thing you need to know. So besides glucose, obviously, what else can you use for energy? I know you can use like muscles and you can use fats, and there's ketones during like when you're fasting, but you can use amino acids. So you can use muscles and protein. You can use ketones, but you can't use long chain fatty acids. Why did I say it like that? Doesn't matter. The only one you can use is short chain fatty acids. And gluconeogenesis is the mechanism by which lactic acid, the Cori cycle, and amino acids, number 1 alanine, are converted to glucose. The only thing you should really know about this in terms of like the Cori cycle using lactic acid is that, long story short, it's like it's a big cycle between the liver and muscles. So the liver, the muscles use glucose to create and then they give off, let's just say, lactate. The liver then takes lactate, goes through the process, the Cori cycle, and then uses that to create glucose. All right, and then the glucose goes back to the muscle creates lactate, the lactate goes to the liver creates glucose, blah, 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 you know the whole shindig, right? But the most important thing you should know in this case is that this is used in times of starvation or stress. And fats and lipids are not available for gluconeogenesis because they break down into acetyl-CoA, and acetyl-CoA it cannot be converted to pyruvate. And if you can't convert it to pyruvate, you can't use it for the Krebs cycle, and what's the point then? You can't create that many ATPs from it. Okay. So you have the other cell organelles, enzymes, and structures and structural components. You have white blood cells. They contain a lot of nucleuses or nuclear material. We know that, obviously, polymorphonuclear cells. You have RBCs and platelets. RBCs and platelets do not have nucleuses. So that's very important. Rough endoplasmic reticulum, it synthesizes proteins. It's mostly found in the pancreatic acinar cells. Not really important, but you should know is it's rough, so it creates proteins. Because the smooth endoplasmic reticulum is smooth, and when you think of something smooth, it's just like fat. It's a blobby. I don't know. You just remember it like that, right? And then this is where where is a big hormone creator of from cholesterol, from fat, from all that stuff. The adrenal cortex, right? When you think about that. All right, so we have we have the Golgi apparatus. Just think about the Golgi apparatus like Golgi G, it looks similar to C, right? So carbohydrates. It just puts a carbohydrate on like the proteins, it tags them with a carbohydrate, And then you have lysosomes. Lysosomes are basically they're digestive enzymes. When they're like small organelles inside the cell that have digestive enzymes that break down any sort of material you want to break down. When the cell, especially when it comes to like white blood cells and such, finds it finds something that's foreign and it wants to get rid of it from the body, it engulfs it, and when it engulfs it, it creates like a border around it, and that border then comes and fuses with the lysosome, and then the lysosome breaks down the material that you want to get rid of, right? If the engulfed particles are small, this is called an endosome. But if the engulfed particles are large, this is called a phagosome, right? It's like phagocytosis, like takes a bite out of it. Anyways, you have the major signaling pathways. You have the phospholipases, the protein kinases, all that stuff. It's a whole other thing. The only one I think you should focus on, or at least you should know according to here, is phospholipase C. And the important part of phospholipase C, if you can just focus with me a little bit, you'll figure this out pretty quickly, right? You have phospholipid phosphatidylinositol 4,5-bisphosphate. Oh, what is that, right? It's like a whole big thing. All that is PIP2. You've heard it before, right? The only thing you should know about it is that when you break it down, it's broken down into through phospholipase C, breaks it down into DAG or DAG and IP3, right? These serve a common purpose, but they serve it through different pathways. The DAG activates protein kinase C immediately. On the other hand, IP3 causes the release of calcium from the smooth endoplasmic reticulum, and that release of calcium activates protein kinase C. So you have PIP2 or PIP3, PIP2 broken down into DAG and inositol triphosphate. DAG goes straight to protein kinase C, not phospholipase C, protein kinase C, and then IP3 releases calcium, and that activates protein kinase 3. And what's important about the whole thing? When you activate protein kinase C, you phosphorylate other enzymes and proteins that down the cascade, basically. And that's all you need to know. So this is for protein kinase C. So C is from DAG and calcium coming from IP3, right? And then you have protein kinase A, and just think about CAMP, right? So that's it. That's all you need to know, CAMP. You have the MAPK, ERK pathway, I don't know. Honestly, just know that myosin is the thick because muscle contractions. Actin is the thin, it goes above the myo. Intermediate filaments are like the keratin, the desmin, and the vimentin. Keratin is hair and nails, desmin is muscle, and vimentin is fibroblasts. I don't really know how important that can be. Microtubules are important, right? So when you think about microtubules, think about the specialized cellular structures, right? You have cilia, you have axons for the neurons, you have mitotic spindles that we talked about previously when it comes to the prophase and the mitotic spindles happening, and then the metaphase where they line up, the anaphase where everyone's selfish and takes one for their own, and then you have the telophase where the new nucleus comes back. And it helps with the transport of organelles in the cells, whatever. And then that's also part of the centriole, as we talked about. And this is basically cell biology for the ABSITE review 7th edition. This is all you need to know. This is a quick review. And honestly, I'm just making it for myself so I can quickly, like 20 minutes per chapter, get through all of this. I try to explain in more details than you really need, but obviously just make it faster, go through it. Just like the key points I'm mentioning, remember them for the exam, and good luck.